A very good morning to each of you and welcome from ICAR-IIHR, Indian Institute of Horticultural Research. I hope I am audible. Any one of you can respond?
Unmute yourself. Yeah, thank you.
Yes,
Thank you.
definitely.
Thank you.
It is audible, sir.
So thank you. So we from ICAR-IIHR welcome each one of you for this important webinar. So this webinar, actually the title is Novel Ideas of Insect Pest Management, especially in creating entrepreneurship opportunities. So we from Business Planning and Development, that is Agri-Business Incubation Unit of ICAR-IIHR, would like to highlight and give an awareness to each one of you on what kind of entrepreneurship that our institute is developing, especially in plant production aspect and that too in insect pest management. So this is an opportunity for all of you to learn the novel ideas which are of recent origin and have got a major role in plant production aspects. And taking up this entrepreneurship, actually our unit of ABI is actually giving a lot of support in terms of providing the awareness. So mentorship with the experts and the major trainers, and further giving the incubation facility, especially to start your entrepreneurship. And these facilities of both the space and the equipment and the machinery will be provided to you. And further, we are also hand-holding for initial marketing through ATIC, that is Agriculture Technology Information Center of our institute, wherein so we also handhold for the initial 6 months marketing, and if required another 6 months of especially 1 year support for marketing of the product also will handhold. And maybe once you graduate, so this will be totally you will become an independent entrepreneur. And I wish that this particular awareness program will give a lot of enthusiasm among all of you to create that entrepreneurship abilities. And hope even if few of you you come out with these ideas of self-employment, and with that intention, so we start this program. So we are very happy that our director, Dr. B.N.S. Murthy sir, in spite of his busy schedules, say our group discussion on ACRP on fruits is going on, and several other programs are simultaneously are there. With in spite of all these hectic schedules, he made it convenient to be with us to give is the inaugural remarks. I welcome on behalf of all the participants, sir, so to this particular webinar. So I also welcome all our experts. Say the lectures will be given by the eminent scientists of our institute in the division of plant production, especially. So Dr. PVR Reddy sir is there. So he will be talking on ARCA borer control. That is the how actually the formulation made from our institute can control the stem borers in major crops like mango, cashew, then coffee and such kind of things. Madam, actually Dr. Shivananda is there. Dr. Shivananda sir actually has retired from our service of ICR, but still he has made it a point to be with us in this morning, and he will be sharing his valuable experiences, especially with the field level, what kind of interventions it will actually one can go ahead. Then Madam, actually Umamaheshwari, Madam is actually giving on all the biopesticides formulations she has made, and especially for nematode management. So a lot of say things which can be taken up for this enterprise activities are actually available with our nematode management units. Then Dr. Sridhar will be giving highlighting rather on tuta traps, so which specifically say light traps which have been formed, and they have got a major role in successful employment potentials. Then Madam Kamala Jayanthi will be giving on a lecture especially on the aspect of the mealy melt and pheromone traps, so which have a lot of say in actually raising the entrepreneurship abilities in each of you. So these kind of overall we'll have a discussion so finally to clarify some of your doubts and how we can go about for further building the business planning. So with this initial background, I have a great pleasure to start this program. So with the IIHR song, I request Mr. Vinay to kindly play ICR and IHR song together.
जय जय कृषि परिषद भारत की, सुखद प्रतीक हरित भारत की। कृषि धन पशुधन मानव जीवन, दुग्ध मत्स्य फल यंत्र सुवर्धन। वैज्ञानिक विधि नव तकनीके, पारिस्थितिकी का संरक्षण। शस्य श्यामला छवि भारत की, जय जय कृषि परिषद भारत की। हिम प्रदेश से सागर तट तक, मरुधरती से पूर्वोत्तर तक। हर पथ पर है मित्र कृषक की, शिक्षा शोध प्रसार सकल तक। आशा स्वावलंबित भारत की, जय जय कृषि परिषद भारत की। जय जय कृषि परिषद भारत की।
ನೋಡಬನ್ನಿ ಸಸ್ಯ ಸಾಗರದ ಭವ್ಯ ಸಂಸ್ಥೆಯನ್ನು, ಭಾರತೀಯ ತೋಟಗಾರಿಕೆ ಸಂಶೋಧನಾ ಕೇಂದ್ರವು. ರೈತ ಸ್ನೇಹಿ ಪರಿಸರ ಮಿತ್ರ, ರೋಗ ನಿರೋಧಕ ತಳಿಗಳ ಆಗರ. ನೋಡಬನ್ನಿ ನವತಂತ್ರಜ್ಞಾನ ಆವಿಷ್ಕಾರಗಳು, ಮೌಲ್ಯಾಧಾರಿತ ಉತ್ಪನ್ನಗಳು, ರೋಗ ನಿರ್ವಹಣೆ, ಸಾವಯವ ಕೃಷಿ ಎಲ್ಲವೂ ಇಲ್ಲಿಯ ವಿಶೇಷಗಳು. ಟ್ರಾಪು ಸೋಪು, ಕೆಚ್ಚಪ್ಪು, ಸಾಸು, ಜಾಮು ಕೂಡ ದೊರೆಯುವುದು. ನೋಡಬನ್ನಿ ತೋಟಗಾರಿಕೆಯ ಆದರ್ಶವಾದ ಸಂಸ್ಥೆಯನ್ನು. ನೋಡಬನ್ನಿ IIHR, IIHR, IIHR.
Thank you. Now I'll request our director, Dr. BNS Murthy sir, to kindly give the welcome address. Sorry, to kindly give the inaugural remarks for this particular webinar. Sir, please.
Yeah, good morning, Dr. B.L. Manjunath, for having conceived this kind of idea of webinar, novel ideas for insect pest management in creating entrepreneurial opportunities.
Am I audible now?
Yes, sir, audible.
Yeah. Good morning, Dr. Manjunath, Principal Scientist and Nodal Officer, BPD, for conceiving this novel idea of insect pest management, creating an entrepreneurship opportunities webinar. I am very happy to note that more than 200 plus participants have registered, but we see now about 65 of them. In a short time, the others also will be joining. And also we are getting a response from various different kind of personnel here, starting from students to the general public, plus some of those people who are assistant professor and associate professor from the universities, and also KVKs. It's a very good response. Thank you for putting your money and also the time in IIHR to know the recent developments at IIHR, especially in the area of pest control management. Why it is required? If you see the Kisan call center records, that is, you may be knowing there is a Kisan call center that is with 1-800-180-1551 number. About 75 to 80% of the questions what the farmers are asked on the pest control or disease control. In this context, it is very important how to protect our crops. There are different ways of protecting our crops. First, we will see why the pests attack the crop. Any given plant or the crop or even the variety, it has to grow in a particular environment. First, it has the genetic potential and the interaction of environment that makes its productivity measured. G into E interaction, what we say. So here, once the genetic makeup of the plant is not sufficient to protect the crop on its own. The environment, different parameters of the environment, maybe it is a moisture, temperature, wind, any other such things in which the agroclimatic conditions the plant is growing, that makes that particular individual plant to become susceptible for these pests or diseases. So in this context, one aspect is genetics. Here, there could have been one presentation from people who are in the breeding side, especially for vegetable crops, where entrepreneurship opportunities are available to multiply the plants or the varieties for taking up in producing the plants which are resistant or tolerant to pests. This is one aspect. Second aspect is, if at all a pest is attacked on a plant or going to attack, what are the measures that we have to see? Here we are talking about novel ideas. In pest control, we have to use all sorts of techniques: physical, biological, and chemical. These days, people are moving towards both physical and biological control of pests. For this to be a successful thing, we have to have a very good knowledge of what is that particular pest. We start from there. We have to identify. It's just like in a medical field, diagnosis is very important. Here also, diagnosis of which is the pest that is going to attack, or it is attacking that particular crop is very, very important. Second one is understanding the life cycle of that particular pest. How long it is going to stay in different its life stages, maybe from egg, different instars, and also the adulthood and its mobility. These are the things that we need to understand better. At the same time, what are the environmental factors that weaken these different stages? If you know these kind of basic things, that is the scientific part of understanding the pest, then it is easy to identify a particular insect which has the weakness in its own to carry out its complete life cycle. If you know that weakness, we can target that one. There are several means of targeting that kind of pest control measures that is available and identified at our institute, Indian Institute of Horticulture Research. Here we work on about 54 different crops and more than 300 varieties of different fruits, vegetable, ornamental crops are available. Along with that, 150 plus technologies of production technologies, including post-harvest technologies. Even we have the pioneers in developing most of the biological control measures and also physical control measures. These days, you are hearing some of those invasive pests which are very difficult to control, and if we can understand the pest better, we can control these pests. And especially some of these major perennial crops like mango, pomegranate, and even guava and cashew, they are susceptible for a major pest called stem borer. A particular farmer, if he has grown that crop, or even for coconut for that matter, if he has grown for more than 10 years or 15 years, and if you see in front of his own eyes, this plant is getting to a stage of senescence and dry up, it is really pathetic. There are measures to control this kind of problems. If we can diagnose it in the beginning itself and take measures, there are several measures of pest control. I'm happy our team of resource persons, Dr. B.L. Manjunath, who is the brain behind this one, Dr. PVR Reddy, Dr. Sridhar, Hanumanthe Gowda, and Dr. Kamala Jayanthi, and also Umamaheshwari, who is a specialist in nematode control, are here today to give you the different aspects of pest control. Added to that, as icing on the cake, one of our superannuated scientists, Dr. Shivananda, although basically he is a science scientist, he is very much involved in the entomological aspects of pest control. several of the new technologies by out of the box thinking. They have brought in new technologies. With these technologies, we can reduce the pest attack and save the farmers from the loss. And these days, the chemical control is one of those means which is getting least attention because of its own problem. Although there are good chemical control measures, due to abusing of some of those chemicals and limited availability of labels in a particular crop, there is a limitation in chemical control. Although it is most effective means of chemical control, because of environmental pollution also, there is some thinking about moving towards biological control and physical control of these pests. And at this juncture, I thank our organizers for giving me this opportunity, and also for you to go ahead and learn. And this is just the beginning for many of you who are joining for the first time to our institute webinar series. And there are quite a few things to learn from our institute. Our scientists will be always available with you for any of those problems. And I would like to see at least a dozen of you to become incubators here at ABI, so that we can take this initiative forward. Thank you very much.
Thank you very much, sir, for your valuable advice. In spite of your busy schedule, made it a point to be with us this morning and encourage all our participants. Especially, sir has given an overall view of the total insect pest management. So I think that is a good beginning, what we can say in the insect pest management. I think we are definitely hope that in the entire day you will learn a lot of techniques, and this is the starting point from our director. Thank you, sir. Thank you very much for being with us. And so we'll start the next immediate lecture from Dr. PVR Reddy. So Dr. PV Rama Reddy actually is a principal scientist in the division of entomology in the plant production, but basically specialized in agricultural entomology. Sir has worked on various insect pests, and especially so, you'll be talking now on the ARCA borer control, so which is becoming a major problem as a stem borer in major plantations, including cashew, coffee, and especially mango. And nowadays even pomegranate, actually this is being actually tried. So this kind of they will highlight on the aspects of how one can go ahead with the management of this stem borer. Sir will give overall view, and of course it will be followed by the sharing of the field experiences from Dr. Shivananda, sir. Sir, over to you, sir, please.
Good morning, all. I'm happy that so many enthusiasts and budding entrepreneurs have showed a lot of interest to join this particular program and to take up agri-based entrepreneurship. And I'm sure you will not be disappointed, and IHR has got some technologies which are helpful to the farmers at the same time, which can give employment at the same time, entrepreneurship to the interested people. So from it is something like lab to land through entrepreneurs. So here myself, I am an entomologist working on pest management in fruit crops. Here, especially here, it's a very specific talk concerned to one of our technologies. It is called ARCA borer control. Means this is a product, a formulation developed by our institute with a team of scientists. The main inventor, Dr. T.N. Shivananda, as was mentioned, who superannuated, but still he is associated. So it is his idea, and we have evaluated in the field laboratory and found that this is working efficiently for management of these borers. I know most of you, maybe from different backgrounds, may not be knowing much about what is this borer, what are these pests. You know, these fruit trees are vulnerable to different types of insect pests. So in this, especially now, we are concentrating about the mango. There are many more than 100 insect pests, but there are only hardly 5 to 6 which are really serious. And out of them, the stem borer is a major one. Means you can see here on this picture, what you are seeing, this like worm type. This is called larva, means immature stage of this borer, which is inside the tree and they eat under the bark. Because of this eating, the tree will die. You can see the right side, the big beetle. This is the adult stage. So same larva, after 6 to 7 months spending inside the tree trunk, it will pupate and emerge as this beetle. So this beetle, by making holes on the tree, they come out. So these beetles come out and again they, after mating, they start egg laying and again this reinfestation. So the cycle goes on. In between you see a mango orchard with trees having some red paint on these trunks. This is the product Arca borer control which is applied to manage this pest. So I will give little more deep insight into this technology. Yeah. So as I was telling, these stem borers, these are one type of beetles. Okay. In Kannada they call Dumbi, and these big beetles in mango, especially old orchards, are more susceptible to these beetles. More than 10 years, and among the what we found, this Alfonso variety, though all varieties get this beetle damage, but Alfonso variety is highly susceptible, and Alfonso is one of the most commercially important varieties. So what will happen, as I told, that beetle generally in May, June, after emerging, they start laying eggs on the main trunk. So those eggs, within a week, they become larva, small worm type. Larva means immature, and they enter into the bark stem. So they go on like this, tunneling inside, and these are the stages which do damage. And sometimes it is not necessary that only one larva will be there. There may be sometimes more than one larva, or even up to 10 larvae also we may find. So because of this continuous tunneling inside, the conductive tissues, means live, which are transport nutrients and water from roots to the leaves tree, they get damaged, and so plant will lose its life supporting system and it will die. Ultimately, you can see the one of the trees completely dried. So see, unlike other insect pests where damage may be seen, and if you don't control, at least next year you will get something. But in case of borers, if you do not take measures, if your 10-15-year-old productive tree dies, see, loss is you are losing, you are going back to again 10 years. So the cumulative loss will run into thousands or lakhs, even depending on your variety. That's why you need to save these trees. So far this management of these borers is bit, means generally we can make out this borer damage only when we see some symptoms of some oozing out of this powder or something. Then farmers, whoever notice, one general practice is injecting some insecticide into the holes so that to kill. Of course, it works in many cases, but what happens, by the time we notice the damage, advanced stage of damage might have happened. If you don't notice, so far there is no effective preventive measure, prophylactic measure. That's why what we thought, no doubt, whatever is damaged through insecticide or through some other physical means, sometimes we taking a screwdriver or some metal hook, you extract the larva and kill. These are the common practices. But what if we protect the tree itself from infestation, just like a helmet giving protection? So some and a safe product, if we can come out, it will be good. So that is the idea, and we, a team of our scientists, as I told, involving Dr. Shivananda and all, we came out with one product, so which is easy to apply and which can be at the same time safe and effective. So that is the product has come out. See, this Arca borer control, it is given name so that ABC farmers can also remember easily. But those who are involved with IHR technologies or mango, this pest, some of you may be aware that our institute only had earlier one product called sealer cum healer. This is also same principle. That was also meant for the stem borer management. But over the years, what happened? It had some limitations, especially the sealer cum healer, that product which was in powder form, that required mixing insecticide, number one. Number two, when it is applied on the trees, it was not withstanding some heavy rains, especially some Malnad region or any rain prone areas. If there is a rain, so it used to, so that was the feedback we got from the farmers. Though it is effective, sir, these are the limitations. So that's why again we sat back and again wanted to refine and improve this. And especially now farmers are going for this organic orchard management, and people are reluctant to use these chemicals. So we thought of having an insecticide free formulation at the same time, which can reasonably withstand this rainfall for a longer period on the stem. So this is the product we have come out by modifying and by mixing some herbal oils. As I mentioned, it does not contain any synthetic insecticide. So it is safe and it will help protecting the trees from this borer attack of any trunk. Though this was evaluated and found, and we are recommending for mango, but many farmers have used it for other trunks like pomegranate, jackfruit, grapes. Even grapes, we have conducted experiments in another research institute in Pune. So in the Solapur area of Maharashtra, there also it has given good results. And Chitradurga district in pomegranate, where in farmers field only in more than 500 trees were applied, and it has worked. And also in Gawa. So means like, because all these trunk borers species is some different, but all belong to one family. So their nature of damage is more or less same. So that is, this principle works for this trunk borers. And what is this novelty of this technology? As I mentioned already, this is a preventive measure, so that if you find some 2% or 3% of trees are affected, and that is the right stage to apply this, so that you can protect your other remaining 80 to 90% of the trees from this. And second thing, it does not contain any insecticides, so it is a safe and compatible with your organic horticulture. And also preparing this material, you do not require any big machinery. See, actually what are the actual ingredients, how to prepare and all will be shared with the license holders. Means whoever is interested in taking this technology, that will be disclosed at that stage. So once this is the in principle, because once you take this technology, you also should know to educate farmers how to apply. See, this is one of our Krishi Vigyana Kendras. They are multiplying it in large scale, and there is a very good demand, and farmers are buying it. So it comes in a 2 kg pack, and it will be in initially in like a slurry type. So you need to dilute it with water. See, about 1 kg can be mixed in around 750 ml, so that to make a slurry, and then you need to apply to the main trunk. See, generally our recommendation is you apply up to maximum 2 to 3 feet or 2 to 2.5, because why we tell that 90% of this stem borer damage occurs only on the main trunk up to the first branch stage. Of course, there may be very rare instances when it goes to the lateral branches, but that is very rare. Generally, dried branches only, they will be attacked by some other borers. So if you take care of this main trunk, you can protect your tree. So this is how it is applied, and generally it has to be applied during May, June, because that is the egg laying period. So this product also has egg deterrence, means once this is applied, the beetle cannot come and lay egg. So it will be repelled. By chance, even if it lays egg, we found that these eggs will not hatch into further next stage. So there itself we are controlling the pest. Instead of again it going inside and again we are inserting chemical, all that steps are not required. So this is the about this product and how to apply. And 1 kg can be applied about 4 to 5 mango trees. Pomegranate means still more trees depending on the girth. And this our institute technology costs license to get license 4 lakh rupees, I think. No. So this is available for any entrepreneurs, and there is a lot of scope for this technology, as many now horticulture is growing, and many farmers are moving from traditional cereal or these crops to fruit crops and orchard crops. And since organic cultivation is on the rise, this product is an ideal component, and this can be one of your business ventures. Means not exactly this, whoever is already having agri based, so you can add certainly this, and you can get this. It is having a good acceptability, and farmers also have given positive feedback wherever it is applied, and through our demonstrations as well as through our KVK studies. So this is what I wanted to share. If you have any doubts, you may ask. Thank you.
Thank you very much, Dr. Reddy sir. Actually, in fact, it is a novel idea of actually coming out with the product, and the details sir has shared with all of you. So it has got a lot of scope in getting the entrepreneurship also. So now you may be having a lot of doubts. So I feel it's the right time, because sir may not be available afterwards. So better you clarify the any points. Let it be any silly issue, no issue. So you please clarify your doubts at this stage.
Good morning,
Good
sir.
morning, sir.
Hello, hold on.
Yep.
1 minute.
Yeah,
One by one, please.
Yeah. This is Saurabh. Hello, am I audible?
Yeah.
Okay, sir. I wanted to know whether can we apply this ABC to coconut trees?
Actually, coconut trees at our institute we have not done, and some people have applied for red palm weevil and other small borers. Yeah, my friend Dr. Hanumanthe Gowda, who works in a Krishi Vigyan Kendra, so he shares his experience regarding the coconut.
Okay, sir.
Good morning, everybody. Actually, as far as the ABC concerned, application of ABC for the coconut, we have not standardized so far. But still, the red palm weevil and others we are not recommending so far. But some pinwall borer is caused by some xylospora species. For that, we are given to that our product is given to many farmers. Actually, they are simply smearing that our ABC into that coconut tree. Actually, so far we are not yet. Actually, we have shared our product to our CPCRA people. Let them come out with the standard recommendation.
Okay, sir.
As on date, it is recommended mainly for fruit trees.
Fruit trees mainly.
Oh, okay, thank you.
Any other question?
Sir.
Hello, sir.
Dr. Prasad. Sir,
Please.
is it for coffee stem borer, sir?
Coffee stem borer also, it was tried in Coorg and it has worked. In fact, we had a collaborative project with Coffee Board.
Yeah, Reddy, can I interact?
Yeah, Dr. Shivananda, who personally taken up studies in the Coorg and Malnad region, he can share.
Yeah, friends, I am Dr. Shivanand. In fact, we have been working on this particular aspect for more than 1.5 decades. And we have successfully come out with a formulation for white stem borer of coffee, that is Xylotropus quadripes. So we had a collaborative program with the Coffee Board, sponsored by Ministry of Commerce and Industry. So we have come out with the organic formulation, which works very, very well for this white coffee stem borer. It has been patented from NBAIR, and the product has been commercialized also, and people have been using in Coorg region. So this product is already available from NBAIR, which is the sister institute of our ICR, and it has been a great success for our institution also. So it works. We have already standardized it.
No, sir. In Mallandur areas are there, sir. The shade management that much it's not there. So there the infestation level was very, very high. there also we can try some demonstrations now, sir. Only in
Yeah, actually, yeah, we can. Why? Because coffee is not our mandate crop, but certainly if any this again, because 3 years Dr. Shivananda and other we all involved and we have taken up already data generated and showed that it is working. But now for demonstration purpose, now actually since coffee is not our mandate, if any request comes from coffee board or any other thing, maybe we will be in a position to provide this material. It can be applied and demonstrated.
yes, sir,
Through
because
Krishi Vigyana Kendras or through coffee board.
because this coffee chamber it is now increasing, sir. There many farmers they are shifting from Arabica to Robusta. So
Yes,
if it is a pronoun technology, we can even our range also we can discuss with coffee board, sir, like that.
No, the technology has been validated by conducting in 30 centers of 3 talukas of Kodagu district. So it has been very well validated and it works. Only thing is somebody has to come forward and then look for demonstrations where the problem is severely seen. So coffee board can coordinate with our institution or with NBIR or with KVK. We as scientists, we will come and we will try to give the guidance and then we will give the all inputs required for the farmers. This much we can do.
No, sir. One more, sir. The moisture level, sir. Due to moisture, the flowering maybe drops in coffee like that. Many farmers they say after applying this, does you find, sir, any flower drop or like that problem?
No. I don't think it's anywhere linked with the flower drop in coffee. See, this is applied on the stem, no. So it doesn't interact with the flowering or flowering behavior or flower drop. So it is purely a mechanical barrier for the insect to come and lay eggs. As Dr. Reddy has pointed out, it works as a barrier.
But commonly the even mass will
Anybody
be
else?
developed now on stem. That moss also that leads to flower drop like that. Many farmers they say, sir. So that's why I'm asking.
No, probably this type of reports we have not seen
Yeah.
because when you apply the stem borer, that moss
No
is removed.
phytotoxicity, no effect on any flower or fruiting.
Yeah.
So far,
Okay.
yeah, it was demonstrated. Anybody else, please?
Hello, sir.
Yeah, yeah,
morning.
please.
Yes, sir. Sir, I have a question. Sir, as I belong from West Bengal, especially in Malda district, you know that mango orchards are there and Fajli Himsagar that is produced. Sir, in their orchard, they actually practice some that you have mentioned that the red color painting, the banding, and somebody may practice some putting some kerosene oil and somebody putting some extra oil. So why they go for ABC? Why they buy? So what to say or what to convince? How to convince them?
Yeah.
What is the advantage beyond this?
Yes,
practice?
a very good question. See, actually, for your information, for last 1 year, we have one network project. Through that, we have given to 20 centers across country, including Malda region and Bihar, Uttar Pradesh, where Dasheri and all. So they are all taking up. See, you are telling them they are banding. See, generally it is a farmer's practice. Many people apply their Bordeaux paste or lime. This kerosene generally not for pasting. They inject it kerosene to kill this.
Yes, sir. Yes, sir.
Okay. As
Yes,
I
sir.
already mentioned, that applying petrol or kerosene or insecticide is to kill the already infested one. Okay. So far, there is no
Yes, sir.
effective product to protect, which is because generally the borer, this behavior is when one tree is affected, it releases some chemicals. Because of that smell, slowly nearby trees and it will spread. And sometimes
Okay.
some orchards may not have borer damage at all. But if you see once borer damage, even 1 or 2 trees, Next, it will go on increase. So how to protect? You have to wait 1 year, and when you see the bole only, then you start injecting kerosene. But here, by applying this product, you are protecting the tree from the beginning itself. No need to wait and to kill the insect through chemicals or other things. And especially this
Okay,
Bihar
sir.
and all, they are all mainly seedling populations and very old trees, and they have more borer problem. So this product will certainly work there.
Yeah, Reddy, can I add a few more points to it?
Yes, yes.
Yeah, Mr. Das, your question was very pertinent. And one of my students has conducted, she has done PhD on this particular topic. What we have found is the organic formulation, what we swab on the tree, will work for nearly 8 to 9 months. Whereas when any conventional chemical, as Dr. Reddy has pointed out, injecting the insecticide or any conventional method will be effective only for about 3 to 4 weeks or maximum 5 weeks. The chemical will degrade and the infestation will start again, and the damage will start appearing within second month onwards. Whereas in our case, in this organic formulation, it will give protection for nearly 8 to 9 months with 90% confidence. That means 90% of the times it will not infect. Of course, when there is infestation in 5 to 10% plants are seen, it can be re-applied and get 100% control measure. This is what is our observation.
Thank you, sir. Thank you very much. I think any of you have got still some more doubts, or we can go ahead.
Yeah, actually our email and all is there. You can provide our anybody. You can contact later also.
So at the end of the session, actually we are providing the mobile numbers and emails of all of our colleagues. So it will be definitely of use to you. You can contact them anytime also. And also you can also interact as and when you get some doubts in between also. So now we'll move on to the next session, that is on field level experiences of ARCA borer control. So I thank profusely, before that I thank profusely Dr. PVR Reddy sir for having come over here and presenting the novel idea of this ABC. So it is definitely, I hope you have actually got the benefit of these things. So anyway, sir will be there. He will interact at the end also in case you have specific doubts. So we'll go with the next session, that is on field level experience of ARCA borer control. Dr. Shivananda sir is already interacted just now with you. So sir has got a lot of practical experience working with the field level farms, many places across the crops on ABC, and of course several other products also. So sir has, although retired, actually he made it a point to interact with each one of you, so that his experiences will be definitely be of much use to you. So on behalf of all the participants, I welcome Dr. Shivananda sir for this program. Sir, the floor is yours.
Yeah. Very good morning, Dr. Manjunath. Thank you for your good words. And in fact, I am really indebted to our director also, who made it a point to mention about the technology which we have developed at IIHR. And I would like to place on record for having given me the opportunity to share my experiences with all our entrepreneurs and as well with other scientists. So good morning, friends. Good morning, everyone. I would like to thank Dr. Manjunath for having given me the opportunity. Now, coming to the point of discussion of today's technology is about sealer cum healer or ARCA borer control. As Dr. Reddy has clearly mentioned that we had already invented a product and we have applied for patent, and we are about to get the patent for the earlier product also. The difference between the earlier technology and this technology is, as Dr. Reddy has mentioned, that this is purely organic, whereas the earlier one was with inorganic. Secondly, this is withstanding the rainfall, and the earlier technology was not withstanding the rainfall. So thirdly, this is sustainable. For 8 to 9 months, you get the protection, whereas in the earlier technology, it was not. And probably we can watch with confidence that no other product in the market is available to give you that much sustained protection for the mango. So this is the reason that we got it patented, and we are about to get the patent for this technology also. So as Dr. Manjunath has been telling, that to share my personal experiences with this technology, I have worked in this particular technology for more than 10 years. And now I would like to share my experiences with the farmers coordination and collaboration. So firstly, in mango, as you all know, that we apply once in a year, maybe much before the rainy season. So with that, you control the borer for about nearly 1 year. So the control is nearly 95%, and you don't have to turn back and then worry about the tree with borer infestation. You are losing nearly 2 lakh rupees loss if it is that if that tree is about 10 to 15 years old, because you will have to calculate to bring up that particular tree for next 15 years, how much money you are spending, how much time you are losing, and how much yield you are losing. So it is better to spend 100 rupees or 50 rupees per tree and then protect your tree in advance. So this has been our sincere guidance to our farmers. So people in and around Kolar district, Tumkur, Chitradurga, and in Karnataka, people have been using and they have been getting very good benefits. So the extent of damage. See, earlier when we were working, myself and Dr. Abraham Varghese, I mentioned to acknowledge the collaboration which I received from Dr. Varghese sir, Abraham Varghese sir. So we both were working together in developing the fruit fly trap for mango fruit fly, Bactrocera dorsalis. So when we were working, the percentage damage of this stem borer was just 2%. Within 10 years, it has come to a stage of nearly 30%. When you walk around in Kolar district, you find lot of orchards are being affected by stem borer, and the people are reporting each and every day that the tree has been lost. So either they partially dry or they fully dry. In order to avoid that, we always recommend that you go for swabbing of this ABC. And now the government of Karnataka has brought into the subsidy scheme, and whomsoever buys our product from the licensed producers of our ARCA, I mean this ABC from IIHR, they get 50% subsidy. So this is another good scheme that the Karnataka government has launched to protect the trees. So probably this can happen even in other neighboring states also. So one gentleman who has already taken the technology from Kadapa, Rainbow City Agrovet Technologies, he has been selling nearly 10 tons of Arca borer, this formulation in his company. So people are benefiting and the farmers are also benefited. So with this, I would also tell that mango is a very highly economical crop which you need to protect. And another very good thing that our Dr. Reddy has told that the technology has entered into All India Coordinated Research Project Trial, AICRP trials. And Dr. Prakash Patil has given this particular product to 21 centers across the country. And 2-year trials are over. And we are so happy to see that the trials wherever they have been conducted, the success percentage is more than 95%. So we are happy that the technology is working. It is working across the country, across the varieties. See, whatever be the variety, it hardly matters. So whether it is Badami or Alfonso or Banganpalli, whatever be it, it works. So with this, I conclude that it works very well in mango. So Dr. Reddy also was telling that about the pomegranate. So people have been raising hue and cry in pomegranate, wherein it is highly intensively cultivated crop. Due to intensive cultivation, the plant becomes susceptible for pests and diseases. This we have to notice it. So because of that, the stem borer in pomegranate is very serious, and the insect is Coelosterna spinator. So this we have studied, and myself and Dr. Reddy, we have conducted field trials in Hirur area, Taluk of Chitradurga district, and wherein we have taken up good number of field trials, and we could control to an extent of 99% with 95% confidence interval. So it's a very good crop, and we can control them, and the people are happy. And now, even now, we have been receiving the calls to manage the stem borer, and we have been successfully managing it. Not only the stem borer, even the short hole borer or pinhole borer, that is also being controlled. And the present practice is to use chlorpyrifos or dichlorvas, which are, of course, dichlorvas is now banned chemical. It is not available, but still some people are using it, which is again not environment friendly. So I would suggest our entrepreneurs to take up this particular technology and try to create awareness amongst the farmers, and then make it organic friendly practices. Now, my friend also has raised about grapes. In grapes also, we have tried in NRC Pune. That is a Psilosterna scabrator. People have tried from NRC Pune, and they have tried for about 3 years. And they have also given the report that it works to an extent of nearly 95% confidence. We can tell that it controls the borer. So it is not only that in mango it has been used, it can be used in pomegranate, it can be used in grapes, it can be used in mango. And one more thing which I would like to mention here is the citrus stem borer, which Dr. Reddy would have forgotten to mention it. This is a very, very serious problem in northeastern hilly region, NEH region of sub-Himalayan India. So the pest is Anoplophora worstigi. So this is again a very serious problem, and we have tried in Barapani in Meghalaya, and the scientists have given very good results that it works to nearly 95% efficacy, and this also can be explored by our entrepreneurs who can take the license from the technology. In addition to this, the jackfruit, see, the Pattocera rufamaculata also affects the jackfruit. Now, jackfruit is the one which is upcoming fruit in South India, especially in Kerala, Tamil Nadu, and Karnataka. It is grown as a highly remunerative crop, mainly on rain-fed fields, that is without irrigation. So this has been a very successful venture in Karnataka, and now many farmers have been coming forth with this particular problem of a stem borer in jackfruit. So we have been suggesting the same thing, and they have been controlling it very effectively. And it's also seen in silk cotton. Of course, silk cotton is not a very commercial crop, but it is grown in Tamil Nadu at few farming levels. So they have also been observing that the whole tree dries, wherein at the base of the crop, the borer attacks and the trunk will dry off. So here also it can be applied and the trees can be saved. And very most important thing is cashew. Of course, Dr. Reddy did mention about this. We have tried this particular technology in cashew also. It is called as root and shoot stem borer. So it is caused by Placoderus ferruginous. So in Tamil Nadu, wherein in TNAU farms, Dr. Ambedkar, one Professor Ambedkar, entomologist, has tried this, and he has conducted trials for about 3 years, and he has given an excellent report that it still works even in cashew also. But the only problem is when it is already affected, we may not be able to retrieve and control the damage, because below the ground, if it goes, it is difficult to apply the material itself. So that's the problem. But still, as a preventive measure, as prophylactic measure, if you take up this swabbing of our ABC on the tree trunk, definitely you will get the control measure. But I am warning you that it should be a prophylactic measure rather than the control measure. So even on cashew, it has been tried, and they have included in the package of practices of cashew also as one of the technologies, successful technologies to manage the shoot and root borer of cashew. Further, I think I have spoken sufficiently enough. So this can be tried in many of the crops. So if any doubts are there, probably you can raise your doubts, which we will be able to answer your points. Thank you very much, Dr. Manjunath, for having given me the opportunity to address our entrepreneurs. Thank you.
Thank you very much, sir. As I told initially, so sir is having a lot of practical exposure, and his experience definitely will be much used to all the field level workers And I'm sure with your experience sharing, many will come out with the practical ideas of this entrepreneurship. So thank you once again, sir. Now I feel many doubts are further created. So if any of you have got any doubts, come out right now, so that sir may not be available afterwards. Any of you have got any doubts on this ABC? Hello? Please feel free, so that later you don't carry your doubts further. If you let it be a silly question, but better to clarify.
Anyway, my email ID and also phone number will be given by the organizers. So if any one of you would like to interact later also, you are welcome to interact and then get the doubts clarified. Thank you very much, Dr. Manjunath. Thank you so much.
Thank you, sir. Thank you. Thank you very much. So now we will continue this with a small video, which we are at the field level with the help of Dr. Hanumanthe Gowda, our scientist from Krishi Vignan Kendra, Hirehalli. We have made a small video on both production of this ABC and its application. So just for the information of all our participants, we would like to play that a small, of course, it's not a professional video, just for getting a glimpse of the issue. We just made it. So I will request Mr. Vinay to play that video.
Or is also called as a trunk borer. It's caused by Bactocera ramimaculata, is one of the important and prominent one. Earlier it was a very minor pest, now it became a major pest. Wherever we go to any neglected fields, we could able to find at least one tree is affected by this stem borer. The symptoms of this the stem borer affected tree shows the yellowing at the top of the tree, followed by the drying of the particular branch where this grub affects. The entire life cycle is almost more than 360 days. Usually this the grub of the beetle, it starts boring the stem portion just below the bark, and once the insect starts affecting the boring the tree, the phasis comes out. That is the indication of this presence of stem borer. For the management of this stem borer, we need to cultivate the orchard very intensively, and we need to we need to keep the orchard very clean, and we have to grow the at least the intercrops, and the management practices very carefully. For that, our Institute of Horticulture Research came out with one specific product called Sealer cum Healer. Earlier, that this product, which was very popular in between the year 2008 to 2016, just the improved version of one more product called Arca Borer Controller. That's the improved version of that Sealer cum Healer. This product is mainly found at IIHR, and it is extensively we are manufacturing, and we are extensively manufactured here at KVK. After the removal of the grubs from the affected trunk portion, we need to go for the treatment with afterwards, our IIHR product Arca Borer Control, that we need to paste it. For the preparation of
we'll get a glimpse.
Arca Borer Controller, we are using mainly, it can actually
that is a secret.
Consist of the soap oil and other constituents are there. Since it's a patented product, we need not to disclose here. Once you've taken the license, we'll certainly will train you how to manufacture, how to make this product. These are the there are so many steps are involved while preparing this one. Found at IIHR. This is the final product of ABC. Our country is affected with this stem borer. We need to examine this tree very carefully and remove the these grubs, whatever they present in the inside the bark, with the help of a small % EC with 1.5 to 2 ml per liter. This is a very important step. That step will demonstrate it. ಮುಚ್ಚಂದಿರುವನೆಯ 7.5 ml of profinophos. And mix it thoroughly. Mix it thoroughly. Mix it thoroughly. This is the one part. Before that, we need to remove the loosened portion of the bark. And then we need to drench with the entire stem portion. Yeah, like this. Done. ಸಾಕಪ್ಪ. We should fill first cracks and crevices first, and then with the help of brush you have to paste the entire tree. Yeah, first we need to fill the ABC into the entire cracks and crevices, and finally we need to paste like this. This is the final stage of this treatment. Very important and practical aspect of this one. How much quantity each tree is required. It depends on the number of cracks and crevices and the extent of area is damaged. Based on that one only, we need to calculate the requirement of ABC for each tree. For this, for example, this tree is 1.5 kg was pasted here for this tree.
Yes,
Treatment like this. We have to paste this ABC for all the trees especially in the month of December or January, so that we will protect the crop from the damage from the stem borer.
I hope you got a clarity on the application part and how to make this the paste and all those things. So this was really just we thought in the last minute, and our Dr. Hanumanthe Gowda has made it a point to make this video even in a short span of time. I am really acknowledging his services to share his the product preparation and the application to the plant to each one of you. So thank you, Dr. Hanumanthe Gowda, and now we'll go ahead with the next presentation. So next presentation will be from Dr. Umamaheshwari. So Dr. Umamaheshwari, actually, madam is working as a senior scientist in the division of plant production. She is basically a nematologist having a lot of field level experience. So actually, her predecessor, Dr. Rao, was also involved in preparation of several of the biopesticides for nematode management. And in this connection, madam has taken over all the products, and these products are having a lot of field level implications. So maybe the business can run even crores if it can really taken up in a big scale. So madam will share a lot of our ideas how to go about with this preparation and the marketing of these products, especially for nematode management in several of the horticultural crops. Over to Dr. Umamaheshwari, madam, please.
Very good morning to all of you. Am I audible?
Yes, madam.
Okay, thank you. I welcome you all to my presentation on biopesticide production for nematode management. I am Dr. R. Umamaheshwari, Senior Scientist from Division of Crop Protection, IIHR. And what are biopesticides? According to US Environmental Protection Agency, these biopesticides are certain type of pesticides. They are derived from natural materials such as animals, plants, bacteria, and other minerals. And these are majorly classified into three categories like microbial pesticides, plant incorporated protectants, and biochemical pesticides. We are interested about these microbial pesticides, and they are nothing but they are having these products will have the microorganisms like bacterium, fungus, virus, or protozoans as the active ingredient. And the plant incorporated protectants, they are also shortly called as PIPs. They are pesticidal substances that the plants produce from the genetic material that has been added to the plant. Example, the BT cottons. And biochemical pesticides, so they are the naturally occurring substances such as the insect pheromones. They control the pest by non-toxic mechanisms. And why these biopesticides or bionematicides? What is the need for going for these biopesticides? And before going to that, we will have a general introduction about nematodes. Many may not be aware also. These nematodes are nothing but very minute microscopic worms, which are not seen through our naked eye. Where they are very tiny pests. You can see here, these are the minute worms. Through the help of this stylet, they make wounds. They will damage the plant roots. And even though they are very tiny, but they are very mighty pests. And they are major limiting factors for horticultural crop production throughout the world. And on an average, 21.3% crop losses are estimated only through these nematodes. And in major 19 horticultural crops, their damage monetary loss is assessed at 50,225 million rupees in India alone. This was estimated through the All India Coordinated Research Project on Nematodes. And these nematodes not only damage the roots, but also through the wounds, they pave easy entry for other plant pathogenic fungi and bacteria. So this situation is called as the nematode disease complex. And along with nematodes, you can also see other plant pathogenic fungi and bacteria are usually associated in nature. And so this will result in complete crop failure. And for farmers, they are using excessive amount of chemical pesticides. They are dumping so much of chemicals in the soil to control these nematodes. But these are causing various problems to environment as well as human and biotic life. And so now there is a huge demand for the eco-friendly products such as these biopesticides. And here you can see the plant parasitic nematodes, and along with the disease complex of Fusarium disease complex, they are threatening the guava cultivation throughout India. They are majorly, they are transmitted through the infected seedlings. You can see here almost, this is a nursery in Vijayawada, almost 100% of these guava nursery saplings, they are infested with the nematodes. You can see the yellowing leaves and also the stunted growth. And while you are planting these infested nursery seedlings in main field, then you can see within 2-3 years, the entire plant dies because of the nematodes as well as other wilt problems. Here you can see the roots have rotten because of the galls as well as the Fusarium wilt. You can see the blackening of the roots. So the plant dies because of these problems. And how these nematodes are transmitted is mainly through the nursery seedlings, as I said, and through the rootstocks, through the grafts, through the ground layers, wherever they are coming in contact with the soil, these nematodes are infesting and they are spreading from the nursery to the main field. And not only in guava, it is also, these nematodes are serious problems in other fruit crops like pomegranate also. You can see the yellowing, and in the roots you can see the gall symptoms. This is a typical symptom caused by this root knot nematodes, which is a major nematode in almost all the horticultural crops. And here you can see the vegetable crops, especially the carrot, beetroot, sweet potato, chilies, tomato, almost all vegetable crops are prone to this nematode attack. You can see even the qualitatively also it is affecting not only the quantity of and reduced size, but also because of these galls, they are not fetching enough market value. And how to diagnose this problem? First, initially you can see in the center of a picture, you can see that nematode damage is starting in patches. Okay, here and there you can see the stunting in patches, and later on these patches, and once the nematode population builds up, you can see the yellowing also, and it spreads to the entire field. And in association with other pathogenic fungi and bacteria, you can see the entire plant dies. And not only in open fields, but also in polyhouses, these nematode problems are very severe. You can see whatever crop is grown in the polyhouses like tomato, chilies, capsicum, gerbera, carnations, cucumber, these are all prone to nematode attack, and nematode problems are very severe in polyhouses because of the conducive climatic condition inside, high temperature and high humidity and more agronomic inputs, and also due to the longer duration of the crops and monocropping, nematode problems have become very severe in these polyhouses. And what are the biopesticides available? In India, under the Central Insecticide Board and Registration Committee, that is the CABRC in short, we can say, that is the regulatory authority for registering all these biopesticides. And so far, around 16 biopesticides have been registered under the Insecticides Act 1968. Out of these 16, around 5 has been registered from IIHR. They are these Paecilomyces lacinus, Poconia chlamydospora, Pseudomonas fluorescens, and Trichoderma harzianum and Trichoderma viridi. These 5 are registered from IIHR. And these are the products. Actually, the Paecilomyces lacinus and Verticillium chlamydosporium, these two are majorly bionematicides. They are controlling the nematodes, and especially they target the nematode eggs and juveniles. They are nematophagous fungus. They are the beneficial fungus that affect the nematodes. And in addition, we also have 3 other biopesticides like Pseudomonas fluorescens, Trichoderma viridi, and Trichoderma harzianum. These are all the talc formulations we have developed. And Pseudomonas, Trichoderma, they are these strains are able to control nematodes, as well as they have other fungicidal and bactericidal activity also against the other pathogenic fungi and bacteria affecting the horticultural crops. In addition, we also have another formulation, ARCA plant growth promoter and yield enhancer, that is a combination product of Trichoderma and Pseudomonas. Yes, this is Paecilomyces lilacinus. Now it is called as Purpureocilium lilacinum. This fungus is actually a purple colored fungus. Because of its color, it has derived its name as Purpureocilium and lilacinum. Lilacinum is lilac colored spores it produces. So it is called Purpureocilium lilacinum. It is a bionematicide and it is a nematophagous fungus, and it parasitizes the eggs of the nematodes as well as the juveniles by producing the enzyme chitinase and protease. It digests the nematode eggshell, because the chitin is also a component of the eggshell, and so it weakens the nematode eggshell by producing the chitinase enzymes. So the eggshell will dissolve and the nematodes will prematurely abort and they will be killed. And this is another nematophagous fungus. It is Verticillium chlamydosporium. Now the new name is Poconia chlamydosporia. It is also a white colored fungus. It produces chlamydospores. That is why it derives its name chlamydosporium. These chlamydospores are nothing but they can also withstand adverse climatic conditions. And we have Trichoderma harzianum. This is also a green colored fungus. It is having a nematicidal as well as fungicidal activity. And Pseudomonas fluorescens, this is another bacteria. It is a plant growth promoting rhizobacteria, a beneficial bacteria. It is having nematicidal as well as bactericidal activity. And Trichoderma viridi, this is another species of Trichoderma. This is also having nematicidal fungicidal activity. And another product we have ARCA plant growth enhancer and yield promoter. This is a combination formulation of both Trichoderma as well as Pseudomonas. So for disease complexes, it is a very good product. It increases the plant growth as well as it boosts the crop yield. And how we have come out with these biopesticide products? First, we have isolated these cultures from the native rhizosphere or even from the parasitized eggs of the tomato or brinjal plants. From rhizosphere, we have isolated these beneficial fungus and bacteria. And we have identified, and we have through the molecular, both biochemical as well as molecular analysis, we have identified them. And we have tested under lab conditions for their nematicidal activity, for fungicidal activity and bactericidal activity. And those which strains which are having maximum antagonism, that we have selected. And we have developed the protocols for its mass production for its formulation, and also we have seen the shelf life, and we have also tested its bioefficacy and then generated this bioefficacy data from 3 agroclimatic conditions in all over the India in for 2 seasons, and in different crops we have tested, and we have found it very effective. And how to deliver these biopesticides, that is very important, and we have also standardized how to deliver these biopesticides. That is very much important from the farmer's point of view for the success of these biocontrol agents. We can deliver them as seed treatment, substrate treatment, and also soil application, and for standing crop or soil drenching, and all, even along with drip also we can apply this. Here you can see the seeds can be treated with these biopesticides at the rate of 15 to 20 gram per kg seeds. Even the substrates can be treated, that cocoa peat which is used for raising the nursery seedlings, they can be treated with these biopesticides at the rate of 10 gram per kg. So here you can see from the initial stages onwards, we can ward off the nematodes and we can produce disease-free healthy seedlings. And we have also given training to many nursery farmers, and they can even enrich all these cocoa peat. They can treat the cocoa peat with these biopesticides, and from the initial stages we can see that we are producing disease-free seedlings. And next important step is the soil application. Here soil application, we have to enrich these biopesticides in any compost like well decomposed farmyard manure or neem cake or pongamia cake or even vermicompost. These we can enrich. Whatever farmers are applying to the fields, so that farmer manure can be enriched with these biopesticides. Nothing but value addition to this farmer manure. And for 1 ton of farmer manure, what we recommend is 2 kg each of Pasteuromyces, 2 kg of Pseudomonas fluorescens, and 2 kg of Trichoderma. These 3 can be each 2 kg. You can mix with 1 ton of the lot, and you can see here you can see the way nicely it is enriched. The farmers are we have also trained many farmers. Here you can see the neem cake is being enriched. After 1 week, you can see the slight trichoderma are growing out. These filaments are going out, growing in the entire lot. And after 2 to 3 weeks, after you have to keep under the shade, and you have to maintain 20-30% moisture. And after 2 to 3 weeks, you can see the entire lot is ready. And this farmyard manure 1 ton is now enriched with the biopesticides, and microbes have multiplied 1 million times inside this. And 6 kg of biopesticides you applied, and now you can see 1 ton it has been enriched, and this is equivalent to a biopesticide product. And this is very cost effective also. 6 kg and it is multiplied to 1 ton in the farmer's field. And this has to be applied before planting, and this is from beginning onwards we are having the technology. So before planting you have to apply in the field and in the beds where you are going to plant. You can apply this some 10 to 15 days before planting. You can apply all these enriched biopesticides, and 1 or 2 irrigations you can give, and this will nicely decompose in the soil, and the top soil will have very beneficial microbes. And whatever you are going to plant or we are going to sow the seeds, you can have initially very good nice establishment with good germination and good crop stand for the initial phases. But in polyhouses or in perennial crops, you have to continuously grow the crop for longer duration. And so for that also, neem cake is the best component. In this neem cake, you can enrich the biopesticides, and even you can store that in gunny bags for 3 to 4 months. And then and there you can take that 20 kg of this neem cake in 200 liters water in a drum. You can mix it, and even if you can also add jaggery to that to ferment it. You can allow it for 2 to 3 days, and this can be drenched. You can given a soil drenching 2 liters per meter square around the root zone, or even after thorough filtering, that neem cake suspension can be given as through the drip also. So like this, continuously, regularly, we have to apply these biopesticides. And here you can see the Goa farmers, they are all trained and they are using continuously all these neem cake suspensions for reducing the nematode problem. And what is the mode of action? How these biopesticides work? And here you can see the plant root colonization. They have high rhizosphere competency. All our strains nicely colonize the roots, and you can see nice root development. And so because of their colonization, the nematodes cannot invade the root. It prevents the entry of the nematodes. And second, you can also see the egg parasitism. Mainly these are egg parasitic, this Poconia or this Pasteuromyces, they are all egg parasitic. They will parasitize the egg, and so the next generation will be affected. And not only that, they also produce many secondary metabolites and antibiotics And these are all causing the nematicidal activities. You can see the juveniles are all dying because of the active direct contact with these metabolites. And also we have, I have told already that they are secreting this chitinase enzyme. So because of that, you can see the eggshell is dissolved and the nematodes are prematurely, they're coming out and they are dying because of the secondary metabolite production. And not only that, these are all the direct action. Indirectly also, these biopesticides enter inside the plant system. They induce the defense enzymes like peroxidase, polyphenol oxidase, and these enzymes, they will give strength to the plant to resist the nematode and other pathogen attack. So they are also inducing the systemic resistance in the plants. So through all these modes of action, biopesticides are having good effect against the nematodes as well as other pathogens. And what are the advantages? You can see here, they are the plant growth promoters, plant growth promoting rhizobacteria is there. And so plant growth is promoted, and because of their antagonistic action, diseases is suppressed, and also crop yield is boosted around 12 to 15%. Additional yield is recorded wherever these biopesticides are used. And not only that, and they are eco-friendly. This is very important, and they are very eco-friendly products. And for this, Dr. M.S. Rao sir, he was the innovator of all these biopesticide technologies from IIHR. He has received some 8 patents, 7 international patents and 1 national patent for all these biopesticide technologies. And what is the need for industries? Now you know that there is increased public awareness for chemical-free products and residue-free products, and farmers, there is enough awareness, and farmers also have realized the potential for these biopesticide technologies, and there is growing demand for the biopesticides. And how to provide? We depend on industries so that these biopesticides are available to all the farmers in our nation, and it has to be available in every shelf of the pesticide shops. So, but for these biopesticides, there is some regulatory system in India, and for that we have to generate the data, which is very much expensive. Here you can see for registration of these biopesticides in CIBRC, that is Central Insecticide Board and Registration Committee, we have to generate the bioefficacy data for 3 agroclimatic conditions, 2 seasons. That will take around 2 to 3 years. We have to generate the shelf life data. We have to submit the samples to many deposit bureaus, and the toxicology data has to be generated. All these some 10 to 12 years it will take. But now IIHR, for all these 5 organisms, we have generated all this data. It is readily available, and those who are coming to purchase these technologies from IIHR, what is the terms of trade? What all IIHR provides is, that is for permanent registration, for register to get the license from CIBRC. These companies, when they purchase the technologies from IIHR, we provide you the bioefficacy data, that is for 3 agroclimatic conditions and 2 seasons. We provide you the toxicological data and eco-toxicology data and shelf life data, culture deposit details, morphology. These are all required for the registration of CIBRC. All these data copies will be providing you. Along with that, we also sign the MOU. We give the letter of authorization signed by our director, addressed to CIBRC secretary, that you can use our data and our culture slants for commercial production. And also these slants, whatever we provide to you, you have to in turn submit it to ICAR-INBM. This is the National Bureau for Agricultural Important Microbes. There you have to get it molecularly characterized and get a matching report. And we also provide training for 3 to 5 days if required by the clients. We will be providing on the basic culturing, mass production, formulation, quality check. And these are the terms of trade. And there is a tremendous market potential for all these biopesticides. These biopesticides can be used for almost all the crops, not only horticultural crops, even for field crops also. These are tested through ACR nematodes in more than 20 centers. It has been found effective. And also it is highly cost effective. The tag formulations, maybe around 150 to 250 per kg, it can be sold. And the biopesticide technologies, whatever is commercialized from IIHR, so far we have more than 550 industries. We have sold these technologies all over the nation. Bacillomyces, Trichoderma harzianum, Trichoderma viridi, Pseudomonas, Arca plant growth promoter, and yield enhancer. All these we have sold to so far more than 550 industries all over the nation. You can see our technology has reached, and we have also recorded many success stories. Here is one example of a guava farmer, and he has a big nursery. He has even purchased purchased our Arca-Kiran technology, is producing guava seedling, but he was facing severe nematode problems, and thousands of saplings have been infested with nematodes, and he approached IIHR. We intervened with the biopesticide technologies. Now he is regularly using all these biopesticide enriched farmyard manure he is applying, and even this enriched neem cake suspension he is regularly drenching every month, and now nematode problem has decreased, and even he has recorded increase in yield. And this picture has been sent by the farmer himself. He has found that even the wilted guava has recovered by forming new shoots and new roots by using all these plant growth promoting rhizobacterial formulations. And even he is another farmer, a guava farmer. He is also successfully has controlled the nematodes by using all our biopesticide technologies. And this is in polyhouse also. We have seen success by using all these biopesticides. Here, this is gerbera, which is severely, gerbera and carnations are severely affected by nematodes. And this particular farmer, he he got crores and crores of losses, and he approached IIHR and Dr. M.S. Rao and his team. They all intervened with the biopesticide technologies, and now they have controlled the nematodes even without a single gram of carbofuran. They are able to grow the crop, and now they have even expanded their polyhouse from 1 acre to 10 acres. And our ADG and current VC of Dr. Y.S.R. Horticultural University, he also visited this polyhouse, and this farmer has recorded a cost benefit ratio of 1:2.4, and he has successfully reduced the chemicals also by 50 to 75%. He has reduced the chemical usage, and the yield increase was up to 24% by using these biopesticides. And here is another farmer, a floriculture farmer. He is using Arca Prajual technology of tuberose, and tuberose is also highly susceptible to nematodes. Nematodes cause huge losses in tuberose, and he used all our biopesticide technologies. He is nicely enriching his farmyard manure. Even this enriched farmyard manure also he is selling to other farmers, and he is earning around 2 lakhs per acre per month, and per year it is around 25 lakhs in 1 acre he is able to produce. And we also honored him, and we awarded him from IIHR for being a progressive farmers and also taking our technologies, and he is popularizing these among other farmers. And we have also recorded the success stories of integrated insect and nematode management using all these biopesticides and other neem soap, pongamia soap formulations. This we popularized through many TV channels also. We are conducting regularly many training programs. We are creating awareness among the farmers through our extension activities in collaboration with KVKs and agriculture research stations in other states also. We are demonstrating women farmers also. We have covered through various DST and DBT projects. And what is the impact of all these biopesticide technologies? So far, this technology is transferred to more than 550 industries, and we are successfully controlling the disease complex problems in horticultural crops. And these technologies are used in more than 8 lakh hectares, and around 900 crore worth of biopesticides are produced in India, and these are transferred from IIHR. And these are the benefit to could be to the tune of 1,000 crores to the farmers in India. And what are the requisites for the startups for biopesticide production? First, all these they have to register their firm as a small scale industry. They should get the SSI certification, and that firm should be established in an industrial area. They have to apply for local pollution control board clearance. They should apply for GST, apply for trade license. These are the requisites for commercial biopesticide production, and they have to register at Central Insecticide Board and Registration Committee. For this only, we are giving the data as well as the microbial slants. We are giving the complete data package, including the bioefficacy, toxicological data, and the entrepreneur has to purchase the technology from IIHR. This will be the later routed through AgriNovate. This is a single window system at ICAR New Delhi. They will be processing further the purchase procedures. And cost of the technology is rupees 2.5 lakhs plus 18% GST per technology. For one technology, we are charging rupees 2.5 lakhs plus 18% GST. This is without royalty and one-time payment. And for further processing of these purchase procedures, you have to mail to itmu.iihr@icr.gov.in that you are interested in purchasing this technology. Further, we will be guiding you how to proceed. And you can apply for CABRC registration. It takes around 12 to 18 months to get the registration approval. And subsequently, you have to get the marketing and manufacturing license from the state authorities. And the CABS registration certificate is mandatory. Even IIHR provides on-site incubation facilities for the startup entrepreneurs, but the CABS registration certificate is mandatory to enter into IIHR. Thank you.
Thank you very much, madam, for that nice presentation. In fact, madam has made it actually as a entrepreneur only among the rather entrepreneur from IHR side, so that we can sell the technologies to each one of you. So in fact, the guidelines and other things, she made it very clear to you. So any of you are interested for such products, probably I think it's an opportunity to come into this line and take it up in a big way. I hope we have a lot of queries to ask. So madam may not be available later. Please clear your doubts at this stage. Any of the doubts you can ask, whether it is immediate the requirement or the later requirement. Please ask your doubts.
Good afternoon, ma'am.
Yeah, please.
Very thankful to you. Given a nice representation about the biochemicals, bioformulations. I already use Arka neem soap, Arka pongmia soap. AMC is, I get excellent result in my kitchen gardening and other things. So
thank you.
Thanks to all IHR team. And I want to some discussion with ma'am. Give cell numbers or email IDs for further query.
Okay, you can take my number. It is 88.
Okay.
61. 61.
61. Okay.
28. 21.
Okay.
55.
55. Okay, ma'am. Thanks.
Repeat, ma'am.
I will put in the chat otherwise. I will type in the chat.
Okay. Thank you, ma'am. Thank you.
Okay. Thank you.
Excellent representation. Thanks.
Thank you. Thanks a lot.
Thank you. Any other question? Hello? Any other doubts? If you have, please clarify yourself. So if not, I think I am very much grateful to madam for having a detailed picture of the biopesticides and giving a lot of clarity about its the entrepreneurship development also. So on behalf of all the participants, madam, we thank you for
sir. Thank you for this opportunity.
So
anytime you may contact me. I have given my phone number as well as the email ID. Anybody interested in purchasing these technologies can approach me. Thank you very much.
Thank you once again, madam. So we'll go ahead with the next topic, that is on actually the Arka suction light traps. That is normally popularly called as tuta traps. So this presentation actually will be done by none other than Dr. V.Sridhar. So he's a very experienced entomologist in the division of plant production. Actually has contributed a lot to the field, and he has come out with some novel ideas of attracting insects to light traps and tuta trap specifically. And he will share his experiences so that you can take it up on a industry scale for a larger perspective of the technology. Sir, all the participants are waiting for your this one. Kindly take your lecture. So over to Dr. Sridhar, please.
Thank you, Dr. Manjunath, for introducing me for the audience. Very good morning to all the participants. Now we'll have a look at one of the IHR technology on Arka suction light suction trap. Here in the first slides itself, I'll make something clear about this technology. Here Arka is a trademark of IHR, you may be knowing. Here light traps, you might have seen several light traps. Then how it is different, just you will see. And suction light trap. Generally, many of the light traps where the whatever is attracted to the light source, they may not be 100% assured of trapping inside that trap mechanism. That is what is the difference between this one and the what you have already seen. That is one difference. And coming to, we are calling this as initially a tuta trap, keeping in view there is one pest, many of you may be knowing, Tuta absoluta, which is one of the invasive pests which has come to our country 7-8 years back, and now IHR come out with IPM, wherein this light trap is one of the important component for its management, how it can be used and what are the other uses of this light trap, we will see. In this slide, if you see on the right side, if you see, there is a source of light. This is how we have started, because why this light trap concept has come is, it is not a new. Insects are attracted to the light source. How best we can make use of that in an effective way without disturbing the already existing beneficial insects, because majority of the beneficials also may be attracted. That we have to keep in mind. What you are seeing on the left side of this slide is the ARCA 2-tap design. We have started with a earlier with plastic material. Now this is the latest thing, wherein all the components of light trap, like what is the automatic timing system can be arranged. Suppose if you want to keep that light automatically on off during particular 2 hours, 3 hours or 4 hours during night, why it is required? Because each insects have different activity. That is the one thing we have to keep in mind, that is standardized. Those who are interested will give the complete information about any insect. And another thing is, though it is developed for the tuta in tomato, by studying in 1 or 2 months, if you are interested for any other crop, any other specific pest which is attracted to light, by standardizing light, same model can be used for the other pests also. This is what I want to share in the beginning. Coming to the just a brief introduction about what is the use of this light in terms of insects. Though we can see our vision is restricted to the light of VIBJAR, violet to red, that is 400 nanometers to 700 meters, and insects can view this. In addition to this, insects also can see below violet, that is the thing, that is where there is a scope of using the light source with the wavelength less than 400 nanometers. We are trying to have to add some more nanometer range this light spectrum to attract the other insects. That is under progress. This is the, in general, if you see this spectrum, this is what we can see. What we cannot see also insects can see, that is ultraviolet. And what you see in majority of the bulbs which are dim, which cannot give light, they are of the ultraviolet. And what are the typical responses of insects to light? You may be showing some insects are attracted, some are repelled, and different impacts are being taken into consideration while making these light traps. Then how these light traps are being used? Light traps are the one of the best tools for sampling. If you keep a light, you are already aware that about light traps or pheromone traps. If it is something is related to chemical cues, that is the pheromones. If it is a light as a source, means light traps. What they will give? They will give the indication whether any target pest of our interest is in and around our field. That is where these can be used as a sampling tools. And they also play an important role in other than sampling, monitoring. They also help in capturing, killing, and other biodiversity studies also. And one more thing is, in case of light traps, both males and females will be attracted. That is another important point. And automatic light traps are more efficient because these traps do not require farmers to examine all the time. That is the thing. In some of the traps, you might have seen we have to keep water, and we have seen that there is no requirement of any regular maintenance is required. Generally, at a specific time, recommended time, based on our target pest, it will be auto on, auto off. And we have come out with the 3 models. One is without battery, means where wiring facility is there, any farmer's field that can be used, where electricity problem is there, battery operated one, where they are the remote fields, where you can, same technology can be used based on solar energy also. Means where electricity is not available, in those places also they can be used. Based on different sources of light, different types of things are there. These are all the different types of light traps. Coming to the specific to this one, how we have arrived it. For each insect, they have a specific wavelength which is very good. We have evaluated several lights with different wavelengths, around 20 to 30 lights with different wavelengths we tried. And suppose this particular insect, tuta trap, why we are calling it is tuta is an insect. Recently, its name has been changed, Thorimia. That is the name. Its new name is Thorimia absoluta, but let us call as of now it is tuta absoluta. Here, if you see, and at the same time, if you are keeping a pheromone trap and a light trap, in this we have kept in a poly house. What you see here in this container, they are the adults of this tuta insect. Here in this pheromone traps, males will be attracted, whereas in this what we are seeing here, 50% of females, that is in nature. In general, 50% will be males, 50% will be females, and both will be attracted. And here I would like to make it clear whether some of you may be having some assumption that whether any beneficial insects are also attracted. For that, we have to be assured during night time, it is not that we will be running these light traps evening 6 to morning 6. We have to study based on our target insects. What is the peak activity period of that particular insect? During that time only we have to use, so that we are not interfering much with the other beneficials like natural enemies or pollinators. And what is the use of this light traps? How they will be useful is, as females also trapped, egg laying will be reduced, and further damage naturally it will come down. As the population comes down, the number of sprays can be reduced. In one of the trials in tomato, where we demonstrated using these traps, we around 50% of the sprays can be minimized. That is how we have to use. And I want to re-emphasize, this will be a component, one of the best important component in the IPM, integrated pest management, light trap based integrated pest management, how best you can make use of it. Suppose the initial time, suppose tuta, it is very much attracted to the incandescent yellow. That is how we started. A major difference between this ARCA suction trap and others is additionally that suction mechanism will be a major component. Further details will be shared for those who are interested for this technology. Coming to how best this can be used as a technology in IPM, as an example, I will try to share here. This is a light trap based integrated management of South American tomato moth, Tuta absoluta. Here, for those who have not seen, there may be a few people who have not seen, in the market right now, what we have seen, all of you might have observed small holes on tomato. All these small holes are because of this Tuta absoluta. This is typically a leaf miner and also a fruit borer. This is earlier what we have as a leaf miner. It never comes on fruits, it never damages other than that leaves, whereas this particular insect, it feeds not only on the leaves, it comes when flowering starts, it feeds on different parts of the plant and even the fruits. This is what the damage we are observing is, that is because of Tuta. This is the adult what you see in the field. When you use the light traps, this is the insect which is getting attracted, both males and females. That is how we have to integrate it. Close up if you see, this is the damage on leaf. What you see here, just maybe 1 centimeter or less, this size larva will be feeding inside the leaf. And what you see till now, leaf miner, this damage can be seen only on the upper surface, whereas when Tuta damage is there, as it is completely devouring the total chlorophyll starting from upper epidermis to lower epidermis, these symptoms can be seen on both the sides. When reproductive stage starts, it also feeds on flowers and fruits also. That is the major difference. This is the typical symptoms when you are not intervening. And when you intervene using this light traps, how best you can save the crop. This is the case under polyhouse. Similarly, same problem can happen under open conditions also. If you are not taking the IPM measures, this complete crop can go, and when you protect the crop, this can be saved like this. This is the, here using light traps, we will be targeting these adults, both males and females. This is the component, this ARCA Tuta trap. this is kept in the field. This is upside down. It has come that don't here. How best you can use these traps? These light traps, when you are using it at 2 to 3 per acre, based on the severity of the pest, we can reduce the incidence of this insect less than 6 to 8%. That is the based on the demonstration studies it is done, and this will be the important component in the generally recommended IPM for the tomato. As I was telling in the beginning, this particular light trap can be expanded to the other crops also. The only thing, we have to make a different light source, which is the best wavelength, which can attract maximum population of this tuta. This slide depicts integrated pest management, wherein not a single component cannot work. This is where we have used even pheromone traps, even light traps. This is the earlier model. This is the latest model we have come out. Earlier it was plastic type. Now it is metallic one is designed. And if somebody is interested, model can change. The basic principle remains the same. And here, this is the natural enemy in tomato fields where we have to encourage. These are the biological control agents like parasitides. We will be seeing that these natural enemies are not affected by use of these light traps. That is how they have to be in thing. Generally, in general, these light traps can be used for different pests, different crops. Here, what I am targeting is tuta and tomato, but it can be tried for other pests also. It can be targeted. This is the major thing is IHR developed light trap suction method to get rid of the tomato pests. In conclusion, I would like to share that these traps are useful devices to collect various insect pests easily, used mainly for mass trapping, as well as monitoring of the pests, and it plays an important role in IPM. It helps to conserve the ecosystem, and you can make use of taking this technology of suction mechanism being developed at IHR. Thank you. If you are having any specific queries, you can interact.
Hello.
Hello.
Thank you, sir, for nice presentation.
Okay, thank you.
Sir, right time for using the light trap?
Which one?
Right time for using the light trap.
Light trap for this particular tuta, we have standardized it as 8 to 11. That is the best time for tuta absoluta, this particular insect. And it depends. It depends on different. What we have to study is generally based on the target insects. We have to continuously monitor from evening till morning at what is the maximum active period. We have to open that window. That light should be open during that thing. That is the concept here. For this particular insect, 8 to 11 is the good one.
Cost of Arca tuta traps?
Hello?
Cost of Arca tuta traps?
Cost to for licensing purpose, it is 55,000 plus GST. And the cost of individual trap depends on the what is the quantity of the traps we are manufacturing. Suppose if you are manufacturing 100 per unit, cost will be different. If you are manufacturing 30,000, it will be different. The basic version of this one will come down comes to around 4,500 to 5,000. And it is a long lasting. It can come for 2 to 3 years once you take that one. And if it is if somebody wants to with plastic material, still it can be less. But durability is one of the important criteria. Based on that, it can be considered, and it is applied for patent. And the concept, mainly the concept, and what are the light sources required, that will be shared for this insects. But we can work for other insects in other crops also. Thank you.
Sir, what is the cost of this technology, please?
It is 55,000 plus GST.
Sir, hello, sir.
Yeah, please.
Yeah, sir. Can we use any poison bait along with this light trap to test letter trap?
Like, means, can you explain further?
Yeah. That's light trap only collecting for the adult moths, or we can use along with this suction trap poison bait, any poison bait like some
Poison
chemical.
bait. Yeah. Generally, if it is related with some pheromones, suppose next talk is on regarding pheromones. If you keep any pheromone-like material, naturally insects attraction will be more. But poison baiting generally we recommend for the cutworm-like insects, which are generally immature stages, larva purpose. But it can be used in different senses also. Suppose if you are having not only light source based, there are sticky traps also. Suppose same thing we can integrate with some of the light colored sticky traps also. We actually we can use this set for the multiple purposes. Like we can make it design that one for even sticky traps also with different colors. Yes, your idea is good.
Sir, this light trap, we can use any noctuidal family insect, no, sir?
Yeah. That is a thing. That is what I was trying to share. Light traps can be used for any nocturnal insect, and keeping in view to minimize its attraction by minimizing the attraction of the beneficial insects, because during night time several insects will come. That time standardization of the time of on and off, keeping in view our target insects is important, and source of light can be different. We have to use either ultraviolet, or some insects may be attracted to blue light, some may be yellow light. Majority generally comes for yellow light. That standardization of wavelength and the time period when this light trap has to be on and off, that is to be standardized for respect to pests.
Okay, sir. And sir, what is the suction pressure in this trap?
Suction that is being not disclosed, it is more than 1000.
Okay, thank you, sir.
Excuse me, sir.
Yeah.
Hello.
Hello. Please.
Sir, could you tell about the population of tuta? Is it does it come under discrete populations or under continuous populations? And if it comes under continuous populations, which crop stage? At what crop stage should these light traps be installed? Because once the immature stages have gone undermined or have gone in the fruit, what would be the practical purpose for using this light trap?
That is this is good. This I didn't mention. I think I will make it clear. When you are using either pheromone traps or light traps, we have to start from the date of transplanting itself. That is one, because it gives a clear idea about when that incidence is starting. That is the starting of the transplanting is the best time. Even if you are some of the neighboring fields, if tomato is there for monitoring purpose, it should be as early as possible in our main field. That is one. And another thing is whether the population is continuous or discrete type. This particular insect, what you have observed, it is earlier during rainy season it was less, and this is the time when peak incidence will be can be observed up to April, and it is having a continuous population throughout the year, particularly inside the polyhouse. If tomato is there, it is 100% sure that it is surviving. It is not having any generally resting stages, and there won't be any discrete populations. We can see the overlapping populations also. What I mean is, you can see all the stages of this insect. You can see simultaneously eggs, larva, pupa, and adults also. That means it is just like fruit flies. It is having a continuous and overlapping generations also, and we have to keep, whether it is outside the in open field or polyhouses, we have to start using from the initial stage only. Otherwise, that population problem will be there. Anyhow, we will be using the integrated approaches. This should be started from the beginning only, beginning of the crop. And another thing is, we can these light traps we can change easily, just like a we can change from a daily basis also. In different parts of our field we can change, and with the minimum number you can manage as a part of IPM tool. Hope I am clear about your question.
Any other question, please? So if not, we profusely thank Dr. V. Sridhar for having come over here and deliver this useful lecture to all of you and share his valuable experiences. I think it has got a lot of potential. Those of you who are interested, make further contact him. He will share his mobile and email IDs
Yes.
subsequently.
Yeah. Okay.
So that you can get much more information as and when you require. Thank you very much, sir. On behalf
Thank
of
you.
all the participants, we would like to thank you.
This is my mobile. Dr.
Sorry.
Manjunath, you will share the email also. Thank you. Thank you, everybody.
So mobile number has been put in already chat box. You can just note it down. So now we will go to the next and the last session, that is on entrepreneurial opportunities to production of mealy melt, fruit fly and pheromone traps. Madam Dr. Kamala Jayanthi is a well-known expert in the field. Madam is a national fellow also in the field of entomology. So she has come out with a lot of products which are having direct field application and has got a very long innings in getting the expertise of this entomological aspects. So you can ask any type of things, and the products which he has come out has got a lot of say in the pest management in the days to come. So madam, it's over to you.
Thank you, sir.
Yeah.
Thank you so much. Good morning, one and all. Actually, I was asked to introduce you about the fruit fly pheromone traps and also a very new product, mealy melt. I will be talking to you. These are all semiochemical based technologies available at IIHR for prospective entrepreneurs. It is like Arca fruit fly trap, Arca Bacteroplus, Arca dorsolure, Arca cucurlure, Arca agastra and Arca mealy melt. So I will be explaining you what exactly semiochemicals are. So like how we will convey with each other through different languages. Insects do communicate with plants and with their fellow insects through chemical signals. So what exactly it is, insect world is odor world. So what we are doing here, we are trying to identify the critical odors which insects use, and we are using them to confuse the insects so that we can get environmental friendly, effective insect pest management. This is the theme behind this semiochemicals. So how we do it? So we have a state-of-art laboratory at IIHR, which is called GCEAD. It is gas chromatography electroantenna detection. It is something like that, like our ECG only. So what we will take different plant odors or insect odors, and we will try to identify which are really matter to insect through these very detailed GCEAD analysis. So we come about a bucket of technologies which I already showed. So I will be introducing you one by one. The first very important candidate is oriental fruit fly, often called as mango fruit fly. It is Bactrocera dorsalis, scientific name. It's highly polyphagous. Though it is called as mango fruit fly, it has more than 300 fruit hosts, both cultivated as well as wild. So that's why it is very difficult to control this pest, because the population is there all the time. Throughout the year, they are surviving either on cultivated or wild available fruits. So, and another problem is it is a big quarantine problem. If you are into fruit export, yes, it is a very big headache. And there are several species which are just, I call as a sibling species. They are also equally important. So that's what I am telling. These are the several closely related and also economically important fruit flies which attack several fruit crops like dorsalis, correcta and jonata. And if you see, you can see the top figure, very like a needle like, this is the ovipositor of the female mango fruit fly. It just inserts into the fruit and lays the eggs inside the fruit. So many times you can see the exudations like this, latex exudations from affected fruits. Usually it will affect matured fruits only in the field. Slowly the blackening happens around the oviposition area, and rotting takes place. So this is how the mango fruit fly inserts the eggs, and these are the boat-shaped eggs you can see if you closely look into fruit. Later the maggots develop and the fruits start rotting. At this stage we will be very alert about this pest, and so it causes huge losses. And if you see the IPM program, the first point in IPM program is sanitation. This is the very familiar sight of mango field. There will be innumerable fruits falling onto the ground, but many people just neglect them, but that is the breeding site for fruit flies. We established with our data, the fallen fruits are more attractive to fruit flies than fruits on the tree. So actually the fruits on the fallen to the ground attracts the fruit fly. So sanitation plays very important role. So if you see the IPM, sanitation, as I said, it is the major role in management. So you have to destroy all fallen fruits. And also the second thing comes male annihilation using methyl eugenol traps. And also you have to use protein bait splashes. Here I gave the recipe for the protein bait splashes. It should go every week almost. Coming to the male annihilation using methyl eugenol trap, the IIHR actually developed a very cost-effective, eco-friendly parapheromone trap. I am really happy morning one of the inventor, Dr. Shivananda, also interacted with you. This trap was introduced by Dr. Abraham Verghese and further improved upon on various aspects. And you can see here, this is the methyl eugenol trap, which is cost effective and eco-friendly. So it is reusable and very farmer friendly. Even farmer can make at his own farm. He can make. And it is very potential component of IPM across the fruit crops, as I said, because it attacks almost several fruit crops which are cultivated. This IIHR has a patent for this, and there are several firms already licensed and also KVKs. And this is just easy impregnation of male attractant, which is called methyl eugenol onto the wooden blocks. So if you see this parapheromone trap, the license fee is 1.5 lakhs and hands-on training is available for 2,000 rupees and it is non-exclusive basis. This is very powerful component, particularly in horticultural crops where fruit flies are playing a major havoc. Adding to this, we have another Arca-Bactro plus. This is a lure which attracts the both species of Dorsalis as well as Bactrocera correcta. As I said, Bactrocera dorsalis is called majorly mango fruit fly because it comes majorly on mango. Similarly, on guava, there is a fruit fly called Bactrocera correcta, which is often called as guava fruit fly. But we at IIHR researched out the attraction of these both species to both methyl eugenol as well as other attractants, and we found that Bactrocera dorsalis majorly attracted to methyl eugenol, and Bactrocera correcta also comes to methyl eugenol, but not to that extent like how dorsalis comes. So we worked on it and we identified several combinations. This is the just to introduce you to guava fruit fly. It is not only major pest on guava, it also comes on other crops, but usually it is majorly occurs in guava. Methyl eugenol traps, yes, they attract the guava fruit fly, but to some extent. So to improvise, we synergized the methyl eugenol traps with phytosemiochemicals, which is called now as Arca-Bactro plus. So this Arca-Bactro plus is a phytosynergist, as I mentioned you, and it is a potent multi-species attractant. And you need not change your existing setup. If you are already licensed a methyl eugenol trap from IIHR, same you can use, but only you can change the lure. And this is the data which I am giving you. The regular methyl eugenol trap, so you can see the orange bar here. This is Bactrocera dorsalis attracted. This is the Bactrocera correcta attracted. When we made the Arca-Bactro plus, you can see equal attraction. So the trap catches enhances immensely. That is Arca-Bactro plus, exclusively for guava fields, particularly in where the Bactrocera correcta is causing immense damage. So the advantages are attracts multiple species, though we are focusing majorly on correcta. The production cost is very low because very less amount of chemical is used, and the synergistic activity exists with the existing methyl-eugenol, and it is easy to produce. As I mentioned, complements the existing technology without any change in the infrastructure. So again, this is the license fee is 2 lakh, and hands-on training exists and on non-exclusive basis. The third one is Darsoliure Plus, very interesting. And this is a blend which we standardized to attract female fruit flies. As I said earlier, methyl-eugenol, it attracts only male members, not female members. If you see the female fruit fly population in the field, almost even if you catch through methyl-eugenol traps up to 90%, even remaining 10% will be sufficient to fertilize the wild females. That's why initially when I mentioned IPM, I highlighted protein bait splashes. That is to attract and kill female members of the fruit flies also. Here what we did, we developed a recipe to attract female fruit flies, which is again, it is odors from host fruits, and also which elicited highest electrophysiological excitation in females. And this is very eco-friendly. But one thing is, you need to have your own dispenser. We will give you the blend. And so we conducted extensive studies, and also we used latest reverse chemical ecology approaches to formulate this and arrive this blend. And definitely you should have your dispenser to purchase this technology, and that cost is 5 lakhs. Hands-on training exists and on non-exclusive basis. And the third member in another member in our list is Arca-Cucurlure. You must be heard about melon fruit flies which cause havoc in cucurbits, almost all cucurbits. So what we did, usually melon flies are attracted to this what we call usually Q-lure traps. But to be honest, Q-lure is very weak attractant. And so what we did, we collected the volatiles from cucurbit hosts which are highly preferred by the melon fruit flies, and we synergized with the Q-lure. So our Arca-Cucurlure came out. So as I mentioned, melon fruit fly is oligophagous. It comes on all cucurbits, and yes, the pest is active throughout the year, and it oviposits not only on the fruits but also flowers and stems. So your IPM should start quite early before flowering to bring down the pest population. So usually, as I mentioned, for mango fruit fly, the IPM for melon fly also, sanitation takes a big role, and again male annihilation with protein bait splashes. So coming to male annihilation, our Arca-Cucurlure will help you. So we studied all cucurbit crops and we identified the highly preferred crop, and we synergized those volatiles with the Q-lure. So which the outcome of that synergism resulted in Arca-Cucurlure, so more catches. And this is very, as I said, it is eco-friendly and very handy, and very small infrastructure development is needed. And again, already if you have mango fruit fly setup, it doesn't need much. So the cost is 3 lakhs, and hands-on training exists and on non-exclusive basis. Another important thing is Arca mealy melt. If you people are knowing about mealybugs, they are polyphagous, comes across the crops, let it be fruit crops, ornamental crops, or vegetable crops. Major problem, and many insecticides doesn't work mainly because of that mealy coating on the body. You can see here the white mealy coating on the body. So what we did, we developed a formulation which dissolves this mealy coating on the mealybugs. You can see after application of Arca mealy melt, how naked they are. So they just dry due to winds and temperatures, and they fall off. So as I said, mealybugs are very hard to kill. They are polyphagous, and the major problem is mealywax on the mealybugs, which hinders any insecticidal application to reach the insect. That's why they called hard to kill insects. So this is what we did in Arca mealy melt. Just we uncovered the covered. So it exposes the mealybugs. from their protective covering. And yes, you can see here some of our experiments where citrus seedlings were infested with mealybugs. After spray with our Arca mealy melt, how beautifully they recovered. And just I want to show you small video how the Arca mealy melt works. Here you can see this is a beetle vine crop were infested with mealybug. When we sprayed our Arca mealy melt, you can see how it happens. It just dissolves the protective covering of the mealybugs. It's a very beautiful technology, and it can come handy in particularly in urban horticulture where mealybugs are major problem. So entrepreneurs who are interested in urban horticulture can look into this candidate. It is very useful and has huge market potential. So the license fee is 5 lakhs, hands-on training exists on non-exclusive basis. So another interesting thing which is not related to our horticulture crops, but we could develop this in collaboration with the Central Silk Board. People who are from sericulture areas can may have interest into this, because usually you must be knowing the silk moths usually given the brown papers to lay eggs, but the silk moths die within 2 to 3 days, and majority of eggs are retained in the abdomen. So what we did, we developed a formulation called Arca Extra. This is again dependent on the mulberry volatiles. So it enhances the egg release from the female mulberry moths. People from sericulture areas can look into this. And this contains, again, as I said, it is a chiromones from mulberry plant, and it's a beautiful liquid formulation. And when it is sprayed onto the brown sheets, it enhances the egg laying from the female moth. So you can see the percent increase in the egg laying across the treatments. This is the data from Central Silk Board. This is definitely, I can say, booster for silk moths, egg laying booster. And again, the license fee is 5 lakhs and some training exists for this. So after seeing our success in mulberry silk moth, DBT gave us additional responsibilities to work out egg laying, enhancing formulations for 1 year silk moths like Eri silk moth and Muga silk moth. We are working on it. And thank you. Thank you so much.
Thank you so much, madam, for that wonderful lecture. In fact, madam has given a variety of products for purchase to you. So in fact, you have the selection out of which one you go, and what is the requirement for those things madam has clearly brought out. I think you have a lot of questions to ask for the madam. Just kindly come out with your doubts, clear the doubts now itself.
Thank you, ma'am. Nice for the presentation about the pheromone traps.
Thank you.
At on-farm production, I want to try for pheromone traps, methyl eugenol, but I cannot get big success. I use methyl eugenol, this methyl eugenol.
Which company it is?
Yeah, this is the Lomba Chemical Private Limited.
Which one?
Lomba, LOBA Chemical Private Limited.
Yeah. Okay. Actually, methyl eugenol shouldn't make any difference between the companies. In spite of that, if you are really facing problem in attraction using the methyl eugenol, why don't you try with SRL and also Cisco laboratories just to confirm yourself?
Hello.
Yeah, can you hear me?
The voice is very slow.
Can you hear?
Hello.
Repeat.
Yeah, can you hear me now? Am I audible?
Yes, ma'am.
Yeah. Yes. So what I'm telling, actually methyl eugenol from different companies shouldn't make any difference in their attraction otherwise. So anyhow, since you found it is not attracting to the extent it is expected to attract, what I suggest to you, why don't you try the other companies like SRL and Cisco and compare the efficacy?
SRL and Cisco.
Cisco, okay. Yeah.
Okay, ma'am. Any product codes? Which product code you tell me for?
Pardon?
I want. If you know.
Pardon?
Please repeat. You are not that clear.
I know the product code of the methyl of SRL company or Cisco company. If you know, then tell me.
Product
Code. Having some
product code.
code. No, you can refer their company catalogs. There won't be universal catalog number. They will change every year. You can refer to company catalogs. Latest one.
Latest.
This is a product 98%. I use 60.
No, 98. That's what I'm telling. It shouldn't actually differ in the efficacy. But if you are feeling, and also you can give it to for testing for any GCMS lab, they really whether it has anything, because usually even methylusinol doesn't have any expiry date also. So you can work with methylusinol what you procured for a quite long time.
Because this is a very fruitful technology for the
Yes,
farmers.
yes,
Saving the money and heavy losses.
Yeah.
Okay, thank you.
Hello, ma'am. Hello, ma'am.
Yes.
Yeah,
yes,
please.
Yeah. Dr. Das this side. Actually, ma'am, I am working as an assistant professor in Circumstance University in entomology. So when I go for farmers training and all, so farmers are using, you just tell that the milli melt, right? Arca milli melt.
Yes.
Right, ma'am?
Yes.
So they are using the indigenous farmers technology. They are simply using soap solution, and every wax material is gone. And if I have to convince them, so what is the advantage? Because 5 lakh and so much cost is involving. So maximum in case of marginal farmers, they if they want to buy or purchase this product, so we have to concern about their economic because it should be economically sustainable. So what is the advantage? So what is the advantage of Arca millimel? Because they are simply using, ma'am, in case of previously sir told that the herbal oil in case of that fruit butang boda. So they are using that simply kerosene oil or neem oil, or in case of millimelt also they are simply using soap solution, and the wax material washed out. So ma'am, what is the advantage beyond this? Because they have some own technology, and to convince farmer, ma'am, to convince the farmer to adapt the new technology is the very big challenge for us. So how to convince them, ma'am? That is my question.
Let me tell you, I don't underscore the soap solution which the farmers are using, which is traditionally coming with their own knowledge, because definitely soap solution works. Because if your hands are oily, what you will do? You will just wash with soap solution. So I won't. Yeah, I don't undermine the using spraying soap solution, but definitely if there is a point, if soap solution is not working, then definitely we can convince them. But if they are happy and they are getting the control with soap solution, yes, they can go ahead with soap solution. Not exactly they have to go for millimelt, because millimelt does the same job which soap does. But many people won't go for soap solution. That's why we formulated this, and this is totally different from soap, and this is only, it has contact action, and you need not spray the whole plant. Where the mealybugs are there, you have to spray only that patches, because it doesn't have any systemic action. It has to fall on the mealybugs to be effective. But if farmers are really happy with the soap spraying, because that is the traditional method which is coming for so many years, and I don't discourage them.
Yeah, ma'am. Sure, ma'am. Thank you. And what is you just tell that the sustainability they have, Arca millimelt have some contact action. So what is the sustainability? What is the duration, ma'am?
Yes, that is a good question. If they go for soap solution, yes, they may get some results, but which may not be effective on long-term basis. But if they are going for Arca millimelt, which is just totally makes the mealybugs naked and exposes to the wind and temperature, And actually we are advocating this as a spray adjuvant only. But I tell you, the moment you give this one, since they become naked mealybugs, they will just become, they dry off and they fall off. And they are sustainable, and it is the control is for a longer period.
Okay, thank you, ma'am.
Welcome.
Any other question, please? So madam has shown several products, maybe has doubts in each of one of them. So if not, we'll preferably thank Madam Dr. Kamala Jayanthi. She has got a lot of ideas in making the plant production more viable in the present times. And on behalf of all the participants, Madam, from BPD side, we would like to thank you for your
sir.
valuable lecture.
Thank you for giving me this opportunity to interact with these prospective entrepreneurs and also budding scientists. And I am giving my contact details, both email as well as phone. Whoever interested, please contact me. Thank you very much.
So,
yes,
please. Somebody wants to talk? Hello? Even otherwise also, so we will be sharing all the contact details of emails and other things of all of our experts in the chat box also, as well as subsequently to all the participants. And wherever feasible, we will also send the PPTs. I think you can go through and you can just get good ideas, and maybe you yourself can come out with your own proposition, how best the things can be taken further with your entrepreneurship activities. And of course, BPD is always there to guide you further. So now with this, actually all the sessions are over. So I would like to thank all the presenters. But before that, actually, any of you have got any general points to suggest, you are welcome. It can be of any general nature also, or any general issues also. Yes, sir. Hello? Hello? Somebody is talking, please be louder.
Sir, regarding in the bark, when we have some scale diseases, can we apply this the formulation which earlier the scientists have told us?
So, okay, scale insects in bark.
Yeah, yes.
No, probably that will not work. That's what our expert says. So, say because each one will have specific mode of action. Probably for scale insects, you have to use separately.
Okay. Sir, we'll get the recordings, I think.
Yeah, we'll try that. We'll post it to you. No worry.
Right, sir. Right. Thank you, sir.
So, any other questions? Any of you would like to suggest something, or go ahead further? How to go about?
Hello.
Yeah, please.
Am I audible?
Yeah.
Sir, this ABC, what is the best time for applying it on the mango alfonso? And what is after application? What is the duration of the effectivity? For how long it can be effective on the tree?
So regarding the duration, I think Dr. Shivananda has made it clear, 8 to 9 months. So, and what was your first part?
Yeah, true.
Yeah,
but so we have to repeat the application every 8 to 9 months.
Yeah, actually what he told is the egg laying period was in the beginning of the season, May, June, he told. So at that time, if you can able to take care, I think the prevention is better than cure, no? So the effectivity will be much higher. So I think, so application during May, June time, I think it will really prevent. And once it infests, I think he also suggested that there is a possibility of getting other more trees getting infected. So that's why better to put as a preventive measure.
Oh, okay, sir. Yeah, thank you.
Okay. Any other clarifications, please? So in fact, it was actually we thought this was one area from plant protection group wherein it can create a lot of entrepreneurship among all of you. And in fact, we went to bring an awareness in each one of you, so that these are the options available with us. And of course, several people have taken and they have already become industrialists in the real sense from earlier our IHR experience says about it. So if any of you show interest, so we are there to further guide you further. And of course, our experts, they will mentor you to take it up to a newer heights. And initially, as I told you, we will also provide marketing support. And of course, initially the incubation facilities support for about a year, including space and the facilities, equipments, or the machinery, whatever that is required for the initial preparations and selling. Only a requirement is, of course, we have to purchase the license of the technology, whatever that is you are interested. And further, the marketing support for 1 year from our ATIC, where we have a regular the market channel, and we sell a huge quantity of different products developed from IIHR. And all of you are welcome for that kind of entrepreneurship activities. So if no other issues are there, so I think I profusely thank all of you. In fact, we have a wide variety of audience among our this one. Although the number registered was quite high, at least we are happy till now. We have many people have joined in and they have taken the advantage of this our webinar. It is just an entrepreneurship development program to create an awareness among all of you. The products available at IIHR. And of course, any one of you are interested or more details for any one specific product, so definitely you can write to our director, Director ICAR-IIHR, Esarghatta, Bangalore, 89. So, or you can directly send a mail or contact over phone, or you can directly come over here and discuss with directly with the concerned the specialist, and you can take the advantage of the details, and further you can plan your program. So on behalf of all of you, I would like to profusely thank all our mentors here who have made it a point to be with us this morning and made it a very elaborative presentations, especially from the development of the entrepreneurship amongst you. So right from say Dr. PVR Reddy sir, then say Dr. Shivananda, then of course Hanumanthe Gowda helped in video depiction. Madam Umamaheshwari showed many of our products for nematode management. Dr. V.Sridhar specifically taught about the tutor traps, which are very helpful. And finally, Dr. Kamala Jayanthi regarding all the pheromone traps and mealy melt and other related issues. So on behalf of all of you, we would like to thank our presenters, and definitely they will be of much help to you. You can they can take the details from our chat box, the mails or the numbers, and we'll be in contact for further guidance. So with this, we'll wind up our program. We from IIHR, we would like to thank each one of you for your valuable time and having made it a point to be with us this morning, and take a note of all our products. And I personally thank our director also, say who has in the beginning given his inaugural remarks, and in spite of his busy schedule, he made it a point to be with us. And of course, our thanks to all our team of ABI. Mr. Sarthak is here. Of course, our Mr. Vinay is also here. So they are also helpful in conducting this program. And on behalf of the ABI BPD of ICAR-IIHR, we would like to profusely thank each one of you on this occasion. Thank you one and all. We'll close the program right now. Thank you. Thank you.